By the end of this video, you should be able to understand this question. What is the role of inositol triphosphate, or the IP3 pathway? So right off the bat, the first thing that should kick in your head is that, okay, this is inositol triphosphate. That's a secondary messenger. Right, okay. So for a secondary messenger to actually exist, there has to be a first messenger response. And that indeed happens when we have a ligand. This could be a hormone, for example, that binds to a G protein. And this will cause the activation, and this will lead to the production of our secondary messengers right over here. So we have the IP3 and the DAG, and eventually calcium as well. And we're going to see how IP3 is the relationship between IP3 and calcium. We'll see that in a second. Another thing we should be aware of is that the G protein that's involved in this is actually the GQ protein. So why am I telling you this? GS, GI, or the GS protein, the G protein with the GS protein is essentially involved with the CAMP, which is the hunger signal, and GI is basically inhibiting CAMP. So the takeaway from this is that the GQ is the G protein involved in this signaling, and just remember that for exam purposes. So this right over here is a molecule, and this is the phosphatidylinositol 4,5-bisphosphate. And you might be asking me, why am I telling you this? And that's because this molecule is cleaved into our secondary protein, or secondary messengers, excuse me. So we have the DAG and the IP3 that are produced from this molecule. And how is this happening again? Remember, we said that the G protein is activated? This is essentially what's happening. We're going to get to that in just a second. This is a massive overview real fast. And then eventually we have the cell response. And the cell response is to basically phosphorylate target proteins. And that's what this entire signaling was about. When we had the ligand that approached towards the cell, this is what the cell was trying to do. It was trying to phosphorylate a particular proteins that could be metabolism proteins, etc. Our cells are made of proteins. So let's get into the more descriptive part of this, what's happened over here. So the first step we talked about was the first messenger response. And what's happening really is a ligand, first of all, binds to activate the G protein. And remember, we talked about what kind of G protein this is. So once that happens, we have something else, and the phospholipase C. And the lipase should just tell you this is an enzyme, and it surely is. And this enzyme is eventually, the activation will allow the alpha Q to to activate the phospholipase C. And once that happens, phospholipase C is responsible for cleaving the molecule we talked about. Remember, we talked about this just a while ago? It's going to cleave this molecule into DAG and IP3, our secondary messengers. And from this point onwards, it's pretty straightforward what happens. So remember, this is in the phospholipid bilayer. And what then happens is we have the IP3 that's allowed to leave from the phospholipid bilayer, and then it goes to the endoplasmic reticulum. And the endoplasmic reticulum is, we're all aware what that is. So it has these receptors on it, the IP3 receptors. And this IP3 receptor, once it's able to bind, so let's see what happens. Once it's able to bind, so once the IP3 binds with the IP3 receptor right over here, this is going to cause the calcium reflux down the concentration gradient. And we know that there is a higher concentration of calcium inside the endoplasmic reticulum compared to the matrix. So obviously, as expected, it's going down this concentration gradient. The calcium is going to flow down into the matrix, and that's going to cause an increase in calcium concentration as expected. And they're intermediary proteins that are sensing the level of calciums. But that's not the point. That's a different story. So what happens next is this calcium is going to activate the calcium binding kinases, and this CMPK2 or CMPK4, these are now activated, and they're going to go phosphorylate target proteins. But this is not the end. Remember, we talked about that there are two things that are cleaved out of PIP2. It was the IP3 that we talked about right now, and the DAG. DAG is still sitting inside the phospholipid bilayer. And what eventually happens is, with the influx of calcium concentrations inside the matrix, the DAG and the calcium will bind to the PKC. This is another kinase, and this kinase is involved with regulatory other proteins. And once this happens, there's going to be further phosphorylation. So the cell response is now completed. This is the end of it. So quick rundown. we have a first messenger response. The ligand is going to bind with a G protein, and that G protein is going to cause the activation of the phospholipase C, and the phospholipase C is going to cause the cleaving of PIP2, giving our secondary messengers, and that's going to cause, so IP3 is going to cause the calcium to efflux, and the DAG is going to cause the activation of the PKC. So let's test your understanding now. Now, this is a little test on your knowledge. What is the role of inositol triphosphate? So A is saying a peptide hormone receptor on the lipid bilayer. So we know it's not a receptor, it's a secondary messenger, so that's wrong. Transmits a signal from the membrane receptor to the inner membrane. But wasn't that the role of this ligand here? That's right. So this is not right either, so cut that out. Secondary messenger, okay, that's right, for steroid hormones. That's not right at all. Nothing to do with steroid hormones, actually. D is saying secondary messenger that activates calcium channels. Now, this is true because it is a secondary messenger, yes, and the second part is true as well. It does activate the calcium channels, and we saw that over here. It did activate it. It allowed the calcium to flow out. So, yes, that's right. Free nucleotides, we're not even talking about nucleotides. It doesn't have to do anything with that. Used to extend DNA. No, that's not right. So, hopefully you got that right, but obviously that was pretty obvious. It's bolded. But hopefully this video made sense, and now you're able to answer this question. What is the role of inositol triphosphate? I would highly recommend that you go through this. You can find this exact note inside the link over here. It's in the Google Docs. So I would say just go through this, repeat it a couple times, read it, and once it makes sense, just move on.